So the next speaker is Toby Gibson. He's a team leader at Amble. His team works on biological sequence analysis, and he told me explicitly that he will not talk about AI today, but maybe he will inspire some of you to do AI-related COVID research. Welcome, Toby.
Thank you very much. So I'm going to try and get my screen share working, and then start my presentation. So can you see that, Lena?
Yes.
Great. Okay, so I'm going to talk about short linear motifs, which of protein structure to what has just been explained, and particularly the ones that we can see in the ACE2 cytosolic tail. I'm going to need to explain what short linear motifs are first. And I've got a subtitle here: if SARS-2 is hijacking our cell networks, how shall we punish it? Because I'm hoping that we can get insights into perhaps drug repurposing. So we started to look at the entry system of SARS once we were all sent home to our houses in lockdown. And so this is primarily my team members' project, with the addition of Friedrich Rittmann from Merck in Darmstadt and Lucia Chavez, a collaborator in Buenos Aires in Argentina. And as I say, there'll be no artificial intelligence in here. You can think about how you might want to apply it, though, to the situations. So what is a short linear motif? So Tim Hunt, 30 years ago, started to see them in cell cycle proteins and defined them as these motifs are linear in the sense that three-dimensional organization is not required. So they're such short pieces of sequence, they don't fold by themselves. The information content is extremely sparse in the sequences, and that means it's hard for us to predict them bioinformatically. So when we're looking at protein sequences and we think we might have found a candidate motif, we ask, firstly, is it conserved in other species? And then we want to know the protein that the motif is in, and the motif is probably going to bind to another protein. So is that activity going to happen in the appropriate cell compartment? So is the protein of interest in the right cell compartment to have that motif? Then we're very keen on getting proximity data, which is coming increasingly but in interaction networks or in macromolecular complexes. So this is sort of contextual information that we'd like to have to get us interested in candidate motifs. So I'll introduce the motifs from the P53 protein in the genome, and unstructured protein sequence in its N-terminus. I've just been told that I've had an unstable connection, so I don't know if anything got lost here. And there's a folded DNA binding domain. I'm pointing to the representation of the sequence at the bottom, and the DNA binding domain can bind DNA. And in the C-terminus of P53, there's another 100 amino acids of natively disordered structure. I hope you can hear me still. And then we have structures that have been solved of various parts. And so we have these little patches of sequence which are binding to other proteins. And in the C-terminus, you can see that actually the same region is binding to different proteins. Obviously, this means that this is a molecular switch because it can't bind to all of them at once. So having these short, accessible pieces of sequence enables far more interactions of proteins than if they were all fully folded up. So someone should speak if I'm being lost.
This is perfect. So
okay, great. Yeah. Okay, so here's the P53 interaction furball. All of these blobs are individual proteins supposed to be interacting with P53, shown in the center. And while the experimental quality may vary, the fact that so many proteins are considered to interact with it gives some idea of the complexity that we're dealing with in cell regulatory systems. And this couldn't happen if it was a fully folded protein. Okay, so we've known for really quite a few decades now that viruses are using short motifs to hijack cell regulation. Norman Davey, Gilles Trava, and I reviewed this in nearly 10 years ago. And we, in our resource, the ELM resource for linear motifs, about a third of the motif classes are known to have viral examples. So the motifs actually be in the viral proteins. And we have a series of structures, representative structures shown up in the right side, where the small red part is the viral protein, the part of the viral protein binding to a host. So there always seems to be a cellular protein motif which the virus can mimic and hijack. And that's clearly an indicator of the fundamental nature of the cell, that these motifs are so abundant and so much of cell signaling and cell regulation and control of cell state is going through them that viruses can repurpose them for their own ends. Okay, I should quickly mention the SARS-2 life cycle, because the actual protein we're going to focus on is ACE2, which is the host receptor, or the main host receptor for SARS coronaviruses. It's showing here just one ACE2 receptor, but in reality, the many spikes can bind to multiple ACE2 receptors, and each spike can bind to 3 ACE2s. So a process called receptor-mediated endocytosis is initiated by cell surface receptors. The cell surface of our cells are continually resampling and bringing in vesicles. So the virus comes in, gets uncoated, then you have the whole process of it going out. Oops, I'm sorry, I've gone backwards. But we're going to focus now on the ACE2 receptor. I should just say that this is a very simple, oversimplified diagram of vesicle trafficking. And vesicle trafficking systems take very extensive advantage of short linear motifs. So these are ones that are working on different parts of these systems. And remember that the virus comes in through the endocytic system. Right, so we know that most of the ACE2 sequence is folded. I'm not showing the structure here, but the structure has already been solved. The extracellular domains are highly folded. So this plot here is from the IUPred server, which is assessing whether the sequences are likely to be structured or unstructured. So the midpoint of the score, below the midpoint is more likely to be folded, and above the midpoint is more likely to be unfolded. So the little tail at the end of the sequence, this is inside the cell, because this part here, which is showing a very strong desire to be folded, is actually the transmembrane helix, separating their extracellular domains and the short tail. So the tail is predicted to be intrinsically disordered, and it's fairly likely that it is. So we can put just that sequence of the transmembrane helix, which is all hydrophobic residues, and the tail inside the cell, into our server. We're interested in the fact that the motif should be in the cytosolic, so we select that cell compartment, and we want to have the right species, which happens to be Homo sapiens in this case. And then we can set off a search, and it will return a diagram like this. It will give us the IUPred prediction back, and it will give us matches to regular expressions, so text regular expressions. So we have a very simple system for scoring motifs. And as soon as I saw this, I was very interested, because I know that that's a relevant motif, that's a relevant motif, that's a relevant motif. The one I've highlighted, the PTB-APO, is a relevant motif, the SH2 NCK is a relevant motif, and the endocytic target motif. All of these would be interesting candidates. So one thing we went and did is to check in the largest phosphorylation database, Phosphocyte Plus, for evidence of phosphorylation of amino acids. And the most phosphorylated amino acid in ACE2 is this tyrosine residue Y with 13 phosphorylations. These have all come from high throughput phosphoproteomics experiments. So no one has done any low throughput. In other words, no one has done targeted experiments on this tyrosine, but it occasionally appears in data sets of phosphoproteins. And you can see that if we look at across the whole sequence, there are examples of individual reported phosphorylations which are actually in the wrong cell compartment, and this is probably noise. But the strong signal, which is then shown in more detail here, is this tyrosine. So that's interesting for us because tyrosine regulation is often important in endocytic signaling systems. So then we went to check the conservation of these motif signatures, and I will zoom into more detail in a minute. But the top sequence is Homo sapiens, and we have these candidate motifs, which I will go through in a minute. And then we have mammals, we have birds, we have reptiles, bony fish, and then the most primitive or deepest diverged organism in this list, the hagfish, which is a jawless vertebrate, and it has the ACE2 gene and protein, and we haven't been able to find that in any more deeply diverged organism. What is a hagfish? That's a hagfish. Quite common. They don't have jaws. And so 500 million years ago, our ancestors looked like this, and then we diverged from them. Right, let's look at it more deeply then. So we've got 500 million years of conservation of these motifs in these tails. It's one of the nicest alignments of motifs that I've seen. So just wished I'd looked at it a few years earlier. Right, I'm going to go from the C terms, the back end, and introduce these motifs. The PBM, that's a PDZ binding motif. PDZ domains, quite a large family of domains. So we have many copies. They don't all bind the same sequences. They have very strong preference for the very end of the amino acid chain. And only a few of them recognize a TXF motif with high affinity. So our choice of what might be happening here is limited to a few proteins, and that should be resolvable fairly fast. The next motif I'm going to talk about is this NXXF. You can see a red proline. Proline is not allowed because of backbone hydrogen bonding. Now, the known examples of this is another large domain family. PTB stands for phosphotyrosine binding domain. However, many of them don't bind phosphotyrosines, they bind F or Y, so they're not phosphorylated as well. We don't know, again, which member of that family is going to have the preference in here. Okay, oops, I didn't mean to do that. Clicking is a mistake. Right, now, so then I can come over to this rather complicated region where actually we can see 4 motifs superimposed on top of each other. Now the Y is the tyrosine that can be phosphorylated. The only one of these 4 motifs which can be which combine a phosphorylated tyrosine is the SH2 binding motif. So the protein NCK SH2 binding motif has a preference for the kinds of amino acids that are shown here, and the phi is hydrophobic. So it's not an exact residue, but it needs to be a hydrophobic residue. So I'll just go from the top motif there, the I bar motif, this MPY. This MPY motif is known from pathogenic E. coli bacteria, but nobody has ever studied any human proteins with these MPY motifs. The I bar domain it binds to bends membranes. So that's a very practical interaction to have, because you need to bend membranes during endocytosis and other membrane vesicle processes. That MPY is conserved in the hagfish. The reptiles have lost it, but unless you're Harry Potter, you can't ask them why. So, so that's a very nice conserved motif. The endocytic sorting signal is a very simple motif with mainly a Y and a hydrophobic, and it cannot be phosphorylated to bind. But we know already from literature that endocytic sorting signals can switch with SH2 motifs according to phosphorylation. And then the fourth motif is a motif that would dock with proteins that lead to the autophagy systems. And it's known that that SARS and SARS2 assemble, when they're actually assembling the viruses, they are repurposing, for not entirely clear reasons, autophagy components. So this motif would potentially be able to bring the SARS2 protein all the way through to the autophagy systems, and then where the process of replication could begin. So all of these motifs need to be verified experimentally. Just finding them in the sequences doesn't mean that they're real. But in particular, if the autophagy motif does bind, then I think this is going to be very interesting because it opens a path to studying how the virus moves into the autophagy systems. So these are the motifs that we can see in the tail. Right. Now, what about drugging? Well, if we know where we are from by validating, look for targets in the cell that we might be able to hit and therefore slow down the viral infection. Just before I describe these two trials, there are tyrosine kinases like ABL and SARC, which are always needed in endocytosis because they control the formation of the actin filaments underneath the membrane. So we would certainly like to see those targeted in various different kinds of infections. Now here are two tyrosine kinase drug trials that are starting in the UK for COVID-19. And the first one makes very good sense because the AXL receptor tyrosine kinase is a suppressor of innate immunity. So if the virus is blocking, or rather activating it, if the virus activates the AXL activity, then innate immunity will be suppressed to the virus advantage. The second example, and I'm not involved in any way, by the way, these are just published on literature on the internet. So the Bruton tyrosine kinase is an activator of cytokine production. It's basically expressed in white blood cells, and it's particularly the cytokine production in macrophages is an area that they would like to try and target. And in this case, it would be for the late stage of COVID-19 lung disease, where fibrosis is setting in and cytokine storms are happening. So this would be actually treating the symptom rather than the virus, whereas the first study would be looking directly to drug the cell in order to try to suppress viral replication. Okay, so thanks for listening, and I will try to return the screen now.